You enjoyed your wine?
Yes, I graded a lot this weekend. Lots of hours spent grading. You guys will get your exams back on Wednesday. I'm not quite finished yet.
I don't think we're that excited.
So making my way through them. We will spend some time, also plan to spend some time on Wednesday going over the answers. So that's kind of something we'll do collectively as a class. Okay, I'm not posting any kind of key or anything like that. It's sort of a little bit on you guys to understand what it is that you did wrong. So we'll walk through, not in great detail, but we'll spend some time Wednesday walking through what the correct answer was, and then you need to definitely consult your lecture notes, the textbook. ASAP tutors come to me in office hours, make sure that you understand where it is that your logic went wrong if you did get an answer wrong. Okay, because then keep in mind that you're going to see these types of questions again on the second exam and again on the final. Not all completely, because we're also covering a ton of new information as well, but you will receive questions again on the future exam. Okay. Is there any questions for yeah.
You jump curve, that's correct?
No. There's usually not really a reason for it in this class. I know that has happened occasionally in the past. I believe that might have happened from what I hear from Austin when he took it last year. We didn't ever have a need for it. We ran it in winter. When I've run the similar class at UC Davis too, not really. The exams are weighted, so it pretty much takes care of itself. You guys, this first exam was worth 80 points. The next one's 120. The next one's 160. So if you're not learning and improving by the time you hit that second, third exam, then yeah, we might want to have a conversation. But that would be on a personal, by person basis. But yeah, we don't. There's not going to be, unless for some reason you guys, but as of right now, what I have numbers-wise in front of me, you guys are hitting right on the expected mark, which is not to say really high, but I expected you guys to struggle with this first exam. That's really typical in this class, okay? And you guys did. I can see that some work needs to be put into it. But on the whole, it wasn't like everybody got 10 points out of 80 or anything really strikingly terrible. So Tally, did you?
Is this how the grade curriculum, like, is everyone getting it, or?
You guys are right where I expected you, which is, it's definitely room for improvement. There's definitely, I think a lot of you can really, that second and third exam, you guys are really going to be able to ramp it up. And we'll talk more. You guys will have a better idea once you get your exams back, and you can think about your study habits for the class, how much time you're really spending outside of class working problems. I can't emphasize that enough. The only way you can learn this stuff is to practice problems, just like a math class, okay? Work through them. And do them without your notes in front of you. Try giving yourselves these practice exams, essentially, working through homework problems without having the textbook, without having your notes, without having your friends near you telling you the answers. You need to be able to sit down yourself with a pencil and a calculator and understand how to approach the problems. Okay, any other questions? Okay. All right, so we are starting this kind of weird chunk of information, and we're going to try to decide the sequence of these topics for the syllabus and the order that we'll talk about them in. It doesn't always, it's hard to know when to fit everything in. And this whole week pretty much we'll be spending time on bacterial genetics. So this is chapter 7. And it feels kind of odd because we don't revisit bacteria for another few weeks after that. When we talk about gene regulation, we're going to get into the nitty-gritty details of the lactoferron, the tryptophan, the rhabdomyosoferon, and how bacteria regulate their gene expression. But this little segment is more, it's not Mendelian inheritance because Mendel didn't look at bacteria. He looked at the eukaryotic system, he looked at plants. But this is kind of like inheritance patterns in bacteria. some of the same types of concepts that we talked about for eukaryotic systems, like how can we use recombinant progeny to map a gene order? How can we think about transfer of genetic material from a parent generation to an F1 to an F2? You can do the same thing with single-celled organisms, with bacterium, which are haploid normally, right? But we can track their inheritance patterns. It's just a little bit different given the life cycle and the biology behind prokaryotic systems versus a eukaryotic. So this is this little segment on sort of transmission genetics, inheritance patterns, but in bacteria this time as opposed to in eukaryotes. Yeah.
Looking at prokaryotes, are we pretty much focusing solely on bacteria?
Well, viruses actually don't really technically fall under prokaryotic systems. That's like a whole discussion you could have with a virologist alone, whether they consider viruses to be alive and all this kind of thing. We will look at phage viruses that infect bacterium and how that affects the bacterium genetics and how you can track the phage DNA. So if you're thinking viruses in terms of the phage viruses, yeah, we'll talk about them too. Because that's one of the major ways to move. So one of the things we're going to talk about tonight is how you can move DNA back and forth between bacterial cells. And one of the ways we can do that, or one of the ways it happens naturally, and one of the ways you can manipulate in the lab is by using one of those phage viruses to do your work for you. Yeah. Okay. Any other? So this is a whole bacteria genetics kind of section. So for us to talk about bacterium, I know everybody comes into this class with a little bit different background, but we're going to kind of introduce the idea of bacteria as an organism, why it's a good study tool, and some of the ways that you can grow it up so that you can understand in the context of the problems. When you're reading a homework problem and it's talking about Palladium bacterium, you guys know what that means. You can kind of visualize that. There's no lab component for this course, so we kind of have to think about things more theoretically. Okay. But as a model, so bacteria as a model system, okay, they're actually an extremely powerful model despite being a prokaryotic, mostly haploid system, right? There's all kinds of obvious biomedical uses and reasons that we might want to study bacterium. More so, it's actually becoming really pertinent now recently with this huge waves of populations of bacterium that are resistant to antibiotics, right? So this is a big biomedical model still to this day, and of course very much so before the advent of antibiotics. This was a major killer for a lot of people. Simple bacterial infections that nowadays we can treat theoretically with antibiotics, but now we're starting to see strains that are resistant to that. So it's a very, very powerful model in the sense of relating to human health and why humans find things important, right? Genetically speaking, though, why would a bacterium be a good model if we're focusing on the genetic reasons? Yeah, they're cheap. Yeah, they're cheap and they're easy to grow up. A bunch of them. They don't take a whole lot of external resources, nutrients, equipment to grow. They're pretty simplistic. What else about their life cycle is really useful for us? How quickly do they reproduce? Yeah, a really, really fast. They have a super fast generation time. What does that mean in terms of genetics, in terms of thinking about mutants and rare events? Yeah, it's going to happen more frequently. We'll pull out that needle in the haystack analogy. So the fact that bacteria has a really fast generation time and you get millions and millions and millions of progeny pretty quickly means that you can see observed rare mutant events quickly. You can actually see this. So we can actually see those rare genetic events. So you can actually study those rare mutants. And that becomes really important. So we'll talk a lot about all these mutants in bacterium, and it tends to be a misconception that we think about mutants being this really common thing, this happens all the time, and it really doesn't, okay? It's not very common. If any of you remember from your 241 lab, I teach 210 labs, which is the weird new form of 241 and 242, right? And so we run a GFP lab where we're introducing a eukaryotic gene into an E. coli cell. Does anybody remember your transformation efficiency? How efficient was that? Did that work really well, or is it a really small number of millions of bacteria that actually take up that eukaryotic gene? Really small. Yeah, this is I mean, in terms of looking at the full numbers of bacterium, these rare events are still extremely rare. It's just that you're working with an organism that makes millions and millions and millions of them, okay? It's like picking out that one person on the whole planet that has that rare mutational event. So we can actually study those. In terms of growing them, what do bacteria look like? What do we grow them on?
Petri dishes.
Petri, okay, so we kind of think of a solid medium like a Petri dish, right? Okay, so this would be on a solid growth agar. What else? Where else can we grow them? In more of a column in a flask, right? In a liquid medium. Okay. So you can also kind of think about them as being transferable in a liquid growth medium. Okay. Usually in Luria broth, LP. So kind of keeping that in mind that you can see them as colonies on a dish, or you can have them floating around mostly single cell in a liquid form. Another thing is they're actually really easy to transfer genes and genetic material between them. And we can do this artificially, or this can also happen naturally, and so we'll come back to this concept of transferring and introducing new genes. That was my little note up here, ways to transfer DNA between cells. So there's a few different ways that bacteria make this happen, and we're going to go into more detail about what that looks like from a genetic perspective, what the DNA is actually moving around and doing. But it's actually, in the lab, it's very easy to do this, which means you can study all different sorts of things, and we tend to think of, for example, that 241 lab reference I just made with the GFP, what was that eukaryotic gene embedded in? How did you transfer that into your E. coli cells? Does anybody remember? Yeah, it was in a plasmid, right? So you can use things called plasmids, right, and introduce whatever gene sequence you're interested in having, put that in a vector in the plasmid, and then put that into the DNA, or into the E. coli and the bacterium. We usually use markers too. So with bacterium, you guys are sort of become familiar again with a new way to depict, or to the nomenclature for the actual gene names. Okay, we've been going through Drosophila and pea plants and all these different ways to talk about alleles. And so with bacterium, often we have one of the major ones is this idea of, and it's carried often on plasmids, is some sort of antibiotic marker. We'll come back to that concept as well. But be familiar with looking at things like streptomycin, which is abbreviated as strep, and then you can either be resistant to it or susceptible. You got it? If you pull in a cord and the entire row just like falls over. Okay, so we can do this for all different types of antibiotics. Okay, so things like you'll see ampicillin, penicillin, and if you're given a problem, you're usually going to be, you can ask that question during exam if you're confused whether it's talking about a gene allele or an antibiotic resistant. But usually we track bacteria by whether they're resistant or susceptible to an antibiotic, and then also whether or not they can grow on different mediums. Okay, that's another common marker for a bacterium, is this idea, common genes or common mutations that you can follow. I don't know if that's like a very good heading for this little tangential topic, okay? But one of the common mutations is this idea of whether or not, how they grow in their medium. This is a really pretty simplistic organism, right? We're not looking at height of the bacterium, the color of the bacterium's petals on its flower. We can't track those types of phenotypes. So really with a single-celled organism, we look at morphology of the colonies, we look at whether or not they can grow, we look at whether they're resistant to a drug. Okay? So things like the colony, what I just said, the colony morphology, if they grow in some kind of strange pattern or anything. Okay? Resistance to a couple things. Resistance to the antibiotics or medicines, and then also resistance to what Matthew was sort of asking about earlier, the phage viruses. Okay, so some of them are actually resistant to different bacterial viruses that attack them. And then that third one that I have up here on my notes is that idea of how they grow, whether they grow in a certain medium or not. So this is called auxotroph. This term auxotroph is referring to a mutant that needs to be supplemented with some sort of special type of nutrient for it to grow. Okay? So that would be, an auxotroph is in contrast, so sort of verses in your notes, a prototroph. I'll say that again. So prototroph means it can grow on minimal medium. It's wild type, it's sort of considered normal, it's not really a mutant. So this one can grow just fine. A prototroph can grow fine on minimal medium.
So what's an auxotroph?
Auxotroph, you have to supplement some sort of nutrient or substance. So this will not grow. Yeah. So this won't, auxotroph will not grow on minimal medium, whereas a prototroph will. Auxotroph, you have to figure out what type of auxotroph it is by eliminating the different nutrients. Okay? And often these are amino acids, so you'll see things like, for example, you could be a Lu minus auxotroph. And what that's indicating is the gene that metabolizes or uses or creates leucine, you need to actually provide that for this auxotroph for it to grow. You have to give it leucine on its little petri dish plate that it's growing on. You have to provide that in order to see it grow, because that's a deficiency in its growth pattern. It's considered an auxotroph. It's a type of mutant. So it has some kind of mutation in its gene that helps produce leucine or metabolize it. There's lots of different variations on the auxotroph. Yeah, this one would need leucine in order to grow. Does anyone want me to repeat any part of that? Okay. So you'll hear these terms, you'll see terms that are applied often with bacterial genetic studies. This whole idea of isolating these mutants out, okay? So I just mentioned an auxotroph versus a prototroph. So how do you find those auxotrophs? If they're not growing on minimal media, you wouldn't see the colonies even come up until you started adding leucine, or you started adding arginine, or whatever, methionine, whatever the amino acid is, the nutrient that they're deficient in. Okay, so ways to isolate out these mutants. You'll see two different terms. One's called a genetic screen. This idea of screening for mutants versus selection of mutants. Okay, so there's also this concept of a genetic selection. And I want to make sure you guys don't get confused between those two concepts, okay? Because I think, I don't know if the words sound the same or the concepts are the same, so I always see people get confused between when you're screening for something versus when you're selecting for it, okay? And you can apply these terms to eukaryotic systems as well. It's just we see them much more often referred to when we're talking about bacteria. Okay? So So with this idea of a genetic screen, you're looking at a full population of organisms, so a whole bunch of E. coli cells. You're scanning a large number of individuals and you're looking for that one mutant. Okay? The difference here between these two is the genetic screen, you're essentially screening and looking for non-selectable mutants. Okay? So whenever you do a screen, it would be for those mutants that are non-selectable. And you're looking at tons and tons of cells. And by non-selectable, I mean, so the example of the leucine, the oxytroph, that's a selectable marker. Okay, that's a selectable type of mutant. Something like this colony morphology concept here, number one, or if there was pigment, this is more common in fungi actually than bacterium. But if you had some sort of, like an example, if you had a pigmented colonies for whatever reason, that's not something you can select for necessarily. It's just something you would have to plate tons and tons of bacterium and look at all the plates and just see, is there anything that looks weird? Look, there's a purple colony right there amongst a bunch of white colonies. That's weird. Let's pick him out and do something more with him. That would be isolating a mutant that just has some kind of morphology difference, something you can't select for. That's called a genetic screen. You do something like that. So that's just growing up tons and tons of organisms and just looking for the mutant. Okay. In contrast to genetic selection, that's when you're actually applying some kind of selective pressure to the colonies. You're looking for, this would be a selectable mutants is what you're isolating. So that means there has to be some kind of advantage or disadvantage to that mutant. Something like in the case of an oxytroph, there's an advantage or disadvantage. It's either going to grow or it's not going to grow based on the environment that you put it in. Okay. So another example for a selectable marker would be an antibiotic resistance, right? You could easily apply a drug. You put penicillin on your plate. That's selecting for mutants that are whatever antibiotic you pick. So if you put penicillin on a plate, that's selecting for those mutants that are penicillin resistant, right? You're basically killing off all the other populations and only keeping, the only colonies you would see grow on a plate that had an antibiotic are those that are resistant to the antibiotic. Yeah. Okay. This concept of a genetic selection kind of brings up this idea of replica plating. So to understand some of these bacterial genetics problems that we'll walk through, you have to understand that you can take the same set of colonies and move them amongst different plates. Okay, so it's not like you're just stuck with some bacteria on one plate. You can do this concept of replica plating. So you can take your plate of bacterium that has some kind of colonies on it, okay? These are little colonies. And you can have them growing on whatever it is at the time. So say minimal media. You'll often see that term minimal, which means you don't have any kind of extra nutrients provided. It's just the basic stuff for the bacterium. And what researchers do is they actually take like a little dowel that fits the shape of the plate. Okay, it's like a block. And they literally wrap it with velvet. It's a very high-tech system here, this replica plating. So you line this block with some kind of fuzzy substance, right? So it could be something like velvet. And you plop it on top of your plate, okay? So you're doing it in such a way you're not crushing the cells. It's nice fuzzy velvet. Bacteria, I guess, like to hang on. The increase of surface area of the velvet tends to make the bacterium stick. So you take your plate with the colonies on it and your little velvet block and you just blot it straight down on top of the colonies, pick it straight back up, and you have an imprint. You have cells that are stuck to that velvet. And then you take that velvet block and you can then, after sticking it on this plate, you can plop it over onto a new plate. Okay? in this case, maybe you'll start applying there's many different uses for this, excuse me, for replica plating. But in the context of, selection, we could then add an antibiotic to this, for example. You could do minimal plus penicillin. Okay. So all of a sudden you could apply selection to those same population of cells over here, and then maybe now you only get 2 colonies. You're going to get fewer, of course. That should sort of intuitively make sense that once you start applying selective pressures, you're going to have fewer cells. Because now we're looking for those rare mutants that are resistant to penicillin. So in this case, you'd be selecting for these colonies are what? In terms of their genotype for penicillin, what would they look like? What would you write?
Pen R.
Yeah, you'd write pen R. Okay, so the genotype for the bacterium at that loci, or at that locus, sorry, is pen R. They're resistant for penicillin. You're selecting for those colonies that are. And then you can do the same thing. You can keep blotting it with minimal media plus arginine plus leucine. Recover those cells that were leucine minus maybe on your minimal, leucine plus on your minimal media, and then when you blot it onto the plate containing leucine, you can recover those that are mutants. Okay. So this idea of replica plating, you're just taking the same exact colonies, and you can do this over and over and over and over again. And in that fashion, you're testing those cells that have multiple mutant qualities to them, multiple mutant genes or alleles to them. You have to either there's different ways to clean the velvet off, or you can put another padding of velvet so you're not contaminating between. Yeah, you always want to make sure whatever you're touching here, it's gonna still have bacterial cells on it. Yeah, but it's really simplistic. It actually is amazing how efficient this works pretty well. Bacteria are fairly hardy. That's another good thing about them as a model. Yeah. Okay. So that conceptually in a lab, how you would do this makes sense to you guys. And so you're basically taught these are the same cells, the same genetic material. You're just either adding different selective pressures or adding different nutrients, antibiotics, whatever it might be.
So you dip in the velvet block first, and then you're dipping it into the minimal plus the penicillin.
Yeah, or whatever. You can do this a million different ways. So you could make a bunch of different plates that have different properties to them. But essentially it's a way to transfer these cells and move them onto a new growing environment.
Okay.
Yeah. Because otherwise, how would you could, I guess, go with a toothpick or a sterile loop and pick each individual colony and re-stick it. But it's a way to quickly pick up all the colonies, all the bacterium, and put them in a new growing environment. Okay. All right, so how do we transfer DNA back and forth between bacteria? So there's sort of 4 ways that I want you guys to think about. So this is like transmission genetics for bacteria. So from cell to cell, if we start with like a central bacteria cell. So bacteria have their circular chromosome. And they often have, I know we usually depict them with like one little plasmid. They usually, most of the time, they actually have multiple plasmids, okay? So, and for the sake of our little drawing here, we're gonna draw multiple plasmids. So you have chromosome, bacterial genome, and then you have these accessory